Do we have like technical support here? Awesome. Can't see most of the things. Okay, so is everybody here? More or less. Okay, so there's a piece of paper sitting in front of you. And just take a couple minutes and yes, you don't have a piece of paper. Who stole his piece of paper? There's more of them around. Let me bring you. So just take a couple minutes and answer those questions. And try to be short and just write down the first thing that pops into your head or your mind. Any of those two things. Here, sorry. Do you guys want?
Sure.
Yeah. Emmanuel doesn't need one. Okay, just like first thing isn't much better. Just short. Do you have one? You do. Awesome, great. So whenever you have it done, you can just send it over and. Yeah, pass it. Send, or you can email. No, don't put. I mean, you can put your name if you like.
No.
This comes to like. So there's like two types of reviewers. The one don't sign their reviews, and ones they do, and Emmanuel does. And I just don't think that I'm like there yet in my life. There can still my career can still be destroyed very easily. So you can sign this or not. Depends where you think you are and your career of knowledge of phytoplankton. But just, write whatever pops in your mind. And just please write so I can read. Oh, so how are they assessed? That means how do you measure them? What are the method? What are the tools you can use to kind of like gain some knowledge about them? It can be anything. How do you, do you look at them? Do you sing to them? Do you, whatever you think that the best way or the first thing that pops into your head, how would you measure phytoplankton, any aspect of phytoplankton? Thank you. You can already see the European versus US tendency based on numbers of satellites. So we're going to have some. Okay, cool. Awesome. Great. I didn't ask you. Is it okay?
Yes.
I'm sorry. So Mary Jane has a strong opinion of it. Thank you.
Just for a second, could you Cheyenne?
Okay, yeah. You go ahead and you can introduce her now. I think they're still. Thank you.
Just so you know, there's another student from UMaine. There's another student from UMaine here, Cheyenne Adams. She works on a project linking aquaculture, possibly the optical property of non-algal particles in the water. Estuaries around here. So she agreed to come and listen to some of the talks to learn more about the type of optical management that would help.
Aha! I see. Has been signed. Good. Well, I think I'm just going to tape these here, and you guys can come and look at it later, because I can't really read all of them right now. This is a cool one. I'm just going to pull a couple of these. So, okay, so when it comes to the first question, what are phytoplankton or what is phytoplankton? Most of the answers that I can see, oh, did everybody give it to me? No, oh my God, sorry. Anybody else? None? Well, most of the people are answering that they're like microscopic photosynthetic organisms, or that they are drifting particles. That's not correct. They're drifting or drifting plants, and see which is, true. And then there's some interesting things that I found. Wait. Mini floating plants. One of them was really cool. Phytoplankton is not a genetic classification. They are determined based on their lifestyles and shared energy utilization by photosynthesis. So obviously there's different definition based on what your guys' background is. So we can use a definition that kind of says something about their morphology, says something about their lifestyle, says something about the way that they're, their trophic state and the way that they utilize the energy. When it comes to, I'm going to leave this because there's some cool answers. Autotroph, phytoplankton is an autotroph. So how can we access them? How can we measure them? We have quite a lot of remote sensing, and that's where we have the European satellites versus US satellites. We don't have any Asian stuff yet. There's lots of fluorometers, there's lots of HPLC, there's lots of carbon, interesting, POC, chlorophyll A, backscattering, so there's lots of optics. There's some molecular biology. There's quite a lot of tools that all of us can use to talk about the same thing, which is awesome. Some of these tools Mary Jane is going to be talking more about, and we're going to teach you throughout this class. And why are phytoplankton important? Here's one good one that says that they are food for zooplankton. That's a person that I have talked to that person a bit. But that's okay. They're the base of the food chain, and they produce oxygen. That's good. They're the primary producers, the same thing. Foundations of marine ecosystem. They produce 50% of oxygen. Great. So most of the answers are actually saying that they are actually the base of what we know, the life as we know. They are producing oxygen. They are giving us the carbon through the process of photosynthesis that I'm hoping everybody's familiar with. But I'm trying to find some. Usually there's exotic ones. You're just going to have to dig through them. Well, half the oxygen we breathe, that's correct. But the important thing is, as I read this one, all of you have kind of not so greatly similar ideas about what the phytoplankton is, specifically when it comes how do we measure phytoplankton. There's a great diversity in it, and that actually determines how do we see phytoplankton, how do we group phytoplankton. That's kind of what I'm going to be talking about today. This taxonomic approaches to grouping phytoplankton, and how did that evolve through time, and what are we doing in optics to say what phytoplankton is, how is it grouped. And why are phytoplankton important? We back again converge. We know that they're really important for the life on the earth, and I'm going to touch a little bit on that one, but some of the answers are really cool, so I'm going to put them up there. So just generally about phytoplankton, somebody said that they're floating particles. Actually comes from Greek and means floating, they're wanderers. Planktos means to wander, and phyto means it's a plant. And the word is actually quite recent. I thought it's kind of an ancient word or stuff, but it was invented by, it has a name somewhere, a person end of the 19th century. And this is kind of like cool Google Analytics tool that you can use to see the proportion of that word appearing in analyzed Google books. So you can see that, oh, that's really important in our world. It's only 3,000 parts of a percent in all the words. And it had its peak in 1980s. I don't know why. But this kind of tells you that, the importance of phytoplankton is increasing and the abundance of that word is increasing. So when you talk about phytoplankton, we tend to throw these words around. We say, oh, dinoflagellate, the diatom, silicoflagellate, coccolithophore. And what does it all mean? Does that say something about their taxonomy? Does that say something about their morphology? Does that say something about how they live, why they're important? It means all these things together. This is really important to take with you that we are talking about a gigantic group of organisms. They're highly, highly diverse from all these perspectives. So it's in order to determine them, you have to kind of start defining, you have to kind of start wide. So when you talk about phytoplankton, you talk about the organisms that live in an oxygenated environment. These are the organisms that just live in an oxygenated part of the earth, of the water column. You will never find them in anoxic zones. Why? Just because they are the ones who produce this oxygen. Through the process of photosynthesis, they produce oxygen and produce carbon that is then forming as a part, that is the base of the food web. They can do photosynthesis because they have pigments. Pigments that Colin was talking about, and pigments that Mary Jane is going to talk about much, much more. They're usually the ones that we refer single cell particles. Have you guys heard about Sargasso Sea? Why is it called Sargasso Sea? Sargasso? So what is Sargasso?
Micro-floating brown algae.
Yes, to how micro they are. They're micro. They're like patches, gigantic. So they're really not that micro. Macro. Yeah, exactly. They're macroalgae. So looking at the definition of the floating plants, they could be considered kind of phytoplankton, but it's not what we're talking about. Most of the time when we say phytoplankton, we think about single cell organisms. Single cell organisms that sometimes kind of join together in the shape of some kind of colonies. So very often you're going to see these organisms forming these long chains. And it's considered, for example, that by forming these chains, it helps them to orient them in the flow of the water, and therefore maybe the uptake of nutrients is going to be different, beneficial for them. Sometimes they use these puffs, make these puffs or tufts, where then teeny tiny cells seem bigger towards the predators. So therefore predators are not going to come and eat them. These are all like different experiments that people do to look why these particles kind of come together in colonial form. You're going to find some of the cyanobacteria actually form these colonies because the uptake of the nitrogen that they do, by forming colonies, they can separate the uptake of nitrogen in one cell with the oxygenated processes associated with just cellular processes in the second cell. So they just form these colonies, but they're still sitting, we still look at them as a small single cell particles. Quite a lot of them have complex morphology. And you're going to see, as you increase the cell size, the morphology, both internal and external, starts to be much more complex, which tends to be a problem when you're trying to do any kind of optical modeling. And Emmanuel's going to teach you a lot. He's going to have electron beam modeling, and you're going to see that this complexity of internal and external morphology in a phytoplankton cell kind of tends to be a problem. If you talk about just general, the life on Earth as we know it, we can divide it in three. These are like domains. I don't know what is the current is, but we talk about eukaryotes, which are true cells. This is where we sit. This is where the plants sit. This is where most of the thing when you think about life sits. And then you have bacteria and you have archaea. These are not true cells. The reason why they're separated from the eukarya because they don't have within cell structures are not separated by membranes. So their DNA and things just float in there. The difference between these two has to do something with something specific physiology, is also the type of the molecular structure of the membranes. But when you look at all these three parts of the living sphere, we find photosynthetic organisms that live in the ocean in both of these. There are actually archaea that use sunlight for the energy, and they can produce carbon, but those two processes have not been linked together. So they're not dependent on light in such a way that phytoplankton is. But phytoplankton, you can find it anywhere, all the way. So there's a gigantic diversity in phytoplankton. So whoever said that it's not a genetical group, it's just something that we describe based on a way that they live, is a good definition because all these are so diverse. The green algae are much more closer to a carnation than to a red algae. So the genetic diversity is really, really high. So there's many, many different ways of defining phytoplankton. And you can define it, looking at definition of species as a taxa and looking at things associated with it. You can define it as a particle. That's what we like to do in optics a lot. And you can look all these different properties. You can talk about its chemistry. You can talk about the cellular stoichiometry. You can talk about the toxin production for harmful algal blooms. Talk about many things. And also then you can go talking about function. And this function is something that is becoming more and more sexy when it comes to remote sensing, where we're trying to combine the species concept with the function concept. I'm going to touch a little bit upon that stuff. But what you guys are going to be doing here and focusing mostly on is we're going to be talking about the optical properties as, using optical properties to define phytoplankton and using this chemical measurements back to define phytoplankton. But again, taxonomy, that's something that, we have to talk about how to group them. And it all rolls back to this evolution taxa concept, evolution of species. A long time ago, it was actually not 350 years, it's more 400 years, Linné came to this idea of making this concept of taxonomy as we know it now, the name of the species, where you have, the first name and the last name of the species. And that's what we were following for a long time. We still follow that, where the classification of the phytoplankton, as any other organisms in the world, was based on morphological characteristics. And since these guys are microscopic, we only had light microscopy to talk about, no? So we would take species like this. This is a dinoflagellate, this is a diatom, and people would really spend lots of time to describing miniature morphological characteristics of each of these algae so we can define them as species. And we took that with us. Like 40 years ago, 50 years ago, we started using, or maybe more, we started using electron microscopy. And this electron microscopy was awesome because it gave us a view of internal structure and ultrastructure of the cell. Some of the, I don't have an image here, but some of the coccolithophores have this beautiful coccoliths on the surface, and now you can use this like electron microscopy images and look at the shape of this coccolith to say something which species it is. And then you can use pigmentation. And this method is something that Mary Jane is going to touch upon a little bit more. I'm going to just say something. But once again, rolling to the concept that this kind of looks more brownish than this. So it's the presence of a certain pigments and accessory pigments that allows us also additional taxonomical definition of these algae. So first, if we look at the morphological characteristics, we can try to dump them in a group and define them as particles. We look at their size. That's the first thing. And if you divide the world, the whole world, everything that exists in size, we start from nothing to really big stuff here. 0.2. So that's the magical border of dissolved and real, no? That's what Colin was talking about, everybody's talking about. This is where we filter and we say anything that is small or is dissolved, doesn't have capability of scattering. That's why we don't call it a particle. Does it have really capabilities of scattering or not? No, we're not going to talk about that, but this is where the true stuff starts. So when we talk about phytoplankton, we would say everything from this point to 2 micron is pico. And there's some like sliding definitions here, but that would be picoplankton, picophytoplankton. From 2 microns to 20 microns is nanoplankton. And then from 20 microns on, we enter in the world of microphytoplankton. When we talk about generally plankton, so including zooplankton, we talk about mesoplankton and so on, and really, really big stuff. But we're not going to go there. And although this definition, I was trying to find like historical view of this definition. Mary Jane, I don't know, who came to idea of plopping these things in 2 and 20 and...
Remember, but that's been around since
Always?
probably 120 years.
So way before, we knew a lot about physiological capabilities or ecological niches of all these species. But when you start looking at this species now, this taxa from a concept of what do they do in nature, you're going to find that most of the phytocalcifiers, the species that actually make carbon calcite like shell on them, like coccolithophores, actually sit kind of somewhere in nano range. Some of them are micro, but they kind of sit in this range. Or like the species that produce lots of DMS, dimethyl sulfate, it's a really important atmospheric gas that actually has an impact on weather and cloud formation. They're kind of sitting in this range. If you look at the silica associated phytoplankton, so diatoms, most of them are micro, so there is something to it. So if we start looking at these phytoplankton as a particle and just group them really roughly, just the knowledge about their size can tell us something about what do they do in the environment. Size is not important just for their what they do in the environment. There's a connection, there's a finer connection between those two things. If you think about cell volume to surface ratio, do you guys remember that from like, was it math? What is the relationship between surface and volume? As you're getting larger, what happens? You're getting smaller. So you have you have a round cell, no? And you have a teeny tiny cell that has certain volume and certain surface. And you have a big cell that has a certain volume, certain surface. What is the relationship between those two parameters? Okay, awesome, great. So smaller cells have bigger surface to volume ratio. So now you're a cell. Put yourself in the ocean. How do you pick up things around yourself? How do you get nutrients? Yeah, through surface, through diffusion. So it's really kind of cool to have bigger surface to volume ratio because you can get more yum-yums, no? So if you're a smaller cell, there's actually benefit to that because your surface to volume ratio is higher. Therefore, your interaction with the outside world is higher, which is great to get food. There is higher efficiencies of an encounter if you are that way. When you're a smaller cell, there's a bigger chance of you being spherical. So like when we're talking about me theory, we can really apply them really well on pico cells, but as you're getting larger, things become more complex. And that might be the reason just because you're living in a completely different laws of physics are surrounding you, but also it can be just because also not just laws of physics, biology that is around you is pushing you to evolve in that direction. When you're a smaller particle versus bigger particle, the Stokes Law is completely different. So does anybody know what Stokes Law is?
It describes the selling velocity of a particle.
Awesome. So if you're smaller versus bigger, what happens? It just tells you that if you're either way, no, which is great, no, which is awesome. There's a difference, and the difference is such that the smaller particles actually there's more drag, so you're going to sink slower. So which means if you're a smaller particle, thanks to Stokes' law, you're going to be stuck in, much easier is going to be for you to be stuck in a surface layer. For example, diatoms have to like develop different things to allow them to like float in this mixed layer to stay in the euphotic zone. So the size that is associated with the complexity, that is associated with these other things actually allow them for these different ecological niches and survivalships and define their position in biogeochemistry of the ocean. If you look at exposure to light, Colin was talking about that a lot, the difference between small and big cell size. And what's going to happen with that small or big cell when you take it deeper into the ocean and up higher in the ocean, the size will define how that pigments are going to be packaged. The carbon content. If you look at the cell carbon density, so if you plot the density carbon versus the cell size, you're going to see some really striking differences, which is something that will definitely impact the optical properties of that cell. So as you're getting bigger, your cell density is smaller. Why? Most of the big cells are diatoms. Diatoms have this gigantic vacuole in them, which is not filled with carbon. So as you're getting bigger, you're actually getting less cell carbon. So if you're looking at the size, optical, I'm going to talk about it one, not today. If you look at using optics to say something about the size, try to derive carbon from it, you have to take that into consideration. Metabolic rates, scale to size. There's different metabolic processes for the small cells and bigger cells, and they differ. So there's nothing real, but you have to take that into consideration. And size, once again, because of all these things, determines carbon export efficiency. So if we look at some kind of like food web, simplified oceanic food web that is highly defined by the size. So here you have a teeny tiny thing, so dissolved, pico, nano, micro plankton, which is phytoplankton, and you're going up here to mesoplankton. You can see that smaller guys have many more interactions than when it comes to the bigger phytoplankton, just because of their size. So when you talk about general processes in the ocean, carbon-associated processes in the ocean, you know when you have phytoplankton community that is dominated by small guys, have more carbon recycling, just because they have more of these interactions happening. But when you have big guys, you have more of a carbon export happening. That's once again associated with the size. So just knowing the size can tell us a lot about, what phytoplankton does and what do we have in the water column. But that's the first stage of morphological characteristics. If we go in a finer stage of defining phytoplankton, then we go in all these Latin names that people throw around. And I'm just going to show you some pretty photos, tell a little bit about it, ask me more. We're assuming here that you guys have some basic knowledge about phytoplankton. Please stop me and just, this is the first time I'm giving this lecture. So cyanobacteria. So this is like in that tree of life, those are the guys that are bacteria. They're not true cells. So what you have the chlorophyll, that is specific chlorophyll, them sitting in these membranes towards the edge of the cell. So this is Synechococcus. It's a dividing Synechococcus. So you find chlorophyll and there are pigments sitting in these like membranes here close to the edge of the cell. Cyanobacteria actually includes most, if not most, of the picoplankton. There's some picoeukaryotes. Many of them do nitrogen fixation. So for difference than other phytoplankton that actually gets the nitrogen from diffusion, these guys are capable of fixing nitrogen from the atmosphere, which is really, really important if you want to do some kind of biogeochemical modeling. They're one of the smallest and definitely most abundant phytoplankton type in the ocean. And their distribution is tropical to cosmopolitan. So they're really, they're really non-dominant in polar regions. Two cool species that you really want to know about, and I'm sure you heard about, are Prochlorococcus and Synechococcus. Everybody knows about coccuses. And these coccuses are really important. They're like dominant, they're the biggest amount of phytoplankton biomass in the ocean. They were just discovered 80s, early 80s, early 90s. Mary Jane will know. And just by accident, because Synechococcus has a fluorescent pigment, phycoerythrin, and that's how they just by accident discovered it. And then later on, using molecular
76 for Synechococcus in 1987.
So me and Synechococcus are almost the same age. So, perspective. But if you look at this, like what I said, this cosmopolitan distribution, Synechococcus is everywhere. But Prochlorococcus we can just find in tropical and subtropical areas. Prochlorococcus is the smallest and most abundant creature in the ocean when it comes to photosynthetic creatures. It's really important. And because it's, go ahead. Did you say something?
It's the most abundant in numbers.
Yes, in numbers, not in biomass. Yeah. I'm talking about abundance in numbers. I mean, it's teeny tiny, You can be, like if an elephant comes, it's going to crush it. But it's really, it's teeny tiny, but it's abundant. It is really important. And we know a lot about it just because we have new techniques. So we have molecular techniques that allows us to detect it. We have flow cytometry that allows us to detect it. But before, when we were just using light microscopy, we didn't know about existence of this creature at all. And they're really dominant, they're really distributed everywhere in the world. So the difference between these two guys, teeny tiny, 0.5 to 0.7. It's a really small size, does not have any kind of phycoerythrin present, but has the vinyl chlorophyll A and chlorophyll B. As you're going to learn, I think Mary Jane is going to be talking about that, bacteria does not have a true chlorophyll A as the rest of the phytoplankton, eukaryotic phytoplankton does have. Synechococcus does have a phycoerythrin, so we can actually use it as a diagnostic tool and in that way discriminate these two species when we're doing usually flow cytometry, because these are teeny tiny, so we cannot see them under the microscope. Trichodesmium, one of my favorite plants, algae. It's cyanobacteria, and it's a nitrogen fixer. So these cells are teeny tiny, but what they do, they kind of clump up together. They make these strings, and these strings, the long chains, form in shape of these so-called tufts or puffs. And it's really awesome because, like, let's say you go on field, you're in Sargasso, and you start filtering for your carbon. One of these puffs are really visible with eye, goes there, and your whole measurement is messed up because there's a gigantic amount of biomass there. And the reason why they form puffs and tufts, there's lots of discussion. And sometimes in a water column, you're going to find a portion of the water column that is dominated by puffs, and a portion of the water column that is dominated by tufts. But it could be like just predatorial reason, or God knows what. They have phycoerythrin, which is a way that we can use it to detect. And that was the way that remote sensing detection of these phytoplankton species was developed. First time. This remote sensing image, this is South Pacific. I looked it up and I forgot. And this is a scale here, this is 20 kilometers. So from here to here is like 200 kilometers. All these squiggly lines here, that's all a trichodesmium bloom. A gigantic trichodesmium bloom. Currently now, I was trying to get an image, but the guy back in the lab who does trichodesmium, he's like off to Barbados or something. There's a bloom that goes from Papua New Guinea, following the whole Great Barrier Reef all the way to South Australia of trichodesmium. It's pushed a little bit by current El Nino situation, but this is a gigantic bloom of this nitrogen fixer, gigantic amount of biomass. It just thanks to phycoerythrin, we can build specific remote sensing algorithms to detect this thing from space. So from there, we're moving a little bit bigger. Now we're going in Premnasiophyceae. I can say this in Croatian, but not in English. Okay, read it wherever you want. But this is the group that has, we're kind of now in nano range. Now nano, maybe a little bit micro, but nano range. This group holds two different big subgroups. First, coccolithophores. Coccolithophores are really sexy, and everybody likes them because they have this really, really cool coccoliths on surface. And these coccoliths are built out of calcium carbonate. And they're one of the most famous phytoplankton, not just because everybody likes them because they're nice and look cute, just because they have a very, very big sensitivity to sea surface temperature. So you can find them in specific ranges. And as such, they were always an important tool in paleontology. Like most of geologists who do paleontology in order to find oil and so on, very often use different species of coccolithophores in order to detect what geological age they are, will they find oil or not. Additionally, there's going to be a paper coming out soon, hopefully, where a student of this class is showing that during the last 60 years, you actually have a movement of these coccolithophores northwards because of the change of the sea surface temperature, which is pretty cool. They have a high sensitivity to pH. So they have calcium carbonate shells. Calcium carbonate, you take it, so you take this and throw it in Coke, it's going to dissolve. And in theory, okay, the ocean acidification will never push the pH so low as that case, but if you coccolithophore and the pH, the solubility of this calcium carbonate is different, it's going to be very hard for you to make this coccolith. So there's quite a lot of work being done on connection between these cells and coccolithophores. They're really important for carbon export. Now, just when they die, they sink and take the particular organic carbon with them, but they take all this inorganic carbon with them down. So they're really good exporters. Again, they're white. They're calcium carbonate. How are they going to look from space? Like that stuff that Colin was talking about yesterday. Now, imagine an ocean doesn't have any absorption, almost minimal absorption, but lots of scattering. And they're really easy to be tracked from space. So Jason's working on that stuff, and his boss, Barney Balch, is going to come here in the 4th week to give a fantastic talk on how to follow these guys in from space and to say something about their biogeochemistry. So these are the coccolithophores. But the next guy, it's a group, it's dominated by species called Cofell Sisters. So these guys do not have these coccoliths in them. However, these are single-cell organisms. They kind of like, when they form colonies, they look like gigantic blobs of goo. There's like a polysaccharide gel where all these little cells are sitting in, and they're just kind of like floating around. They're really big. They're like, I mean, not visible with eyes, but they're really, really big. And when they die, most of this polysaccharide gel, actually, if they start sinking fast, is going to get exported. But most of the time goes in dissolved organic matter. So their presence is actually contribution to dissolved organic matter pool. That's how this organic matter pool gets kind of fed a lot, in usually in the areas where they are dominant, like Arctic regions and so on. They form really ugly film in beaches in UK. I'm sure in some other beaches, but that's where they like really talk about, beaches in UK have ugly film. But that's kind of where they're dominant because of that, all that polysaccharide. You just think about wave action and bubbles it up and makes it really looking ugly. But also when you go to the ocean and you do this big breath, you say, oh, it smells like a sea. So, like when people go in England, I'm not sure Jason can attack me, but people go to, I don't know why they go in England, it's pretty like ugly water there, but you go because of the smell of it. I mean, you have film on the beaches, it looks like Gulf of Maine, but you can go for the smell of the seaside. And the smell of the seaside is going back again that DMS, the metal sulfide, which is highly produced by these Cofell Sisters. So, really, really important group things. And just think about it. Smell of the seaside is actually really bad gas that is changing the climate. So once again, this is a gorgeous bloom of coccolithophore just above Norway. A complete side thing, just kind of stuff. So another postdoc of Barney Balch has a thing with nails. And there's that like stupid thing called Jamboree, which is kind of you buy these stickers and you can put it on your nails. But since she's a geek, she takes this like coccolithophore satellite images, puts them on nails, and you can buy these stickers and walk around with like coccolithophore images on your nails. So a random thing, but kind of like ha ha ha. Okay. I mean, I have some. I'll bring it over. I'm just too lazy to put it on my nails. So now we're going bigger. Now we're going from nano kind of to micro. And again, I'm not going to go through all these groups. There's too many of them. I'm just talking about the most important ones, the ones that we're going to be rolling out like all the time. Dinoflagellates, really, really gorgeous things, beautiful things. The shapes and forms like out of this world. Look at them. This is serratium. These guys look like they're kissing. They just have been divided. No, this looks like a really fiery creature. And they have these great shapes and forms and sizes to, once again, stay afloat, run away from the predation, maybe helps them with feeding and so on. So they have flagellates. They're flagellates containing algae. They're plant-like, but they're sometimes animal-like. So they're sometimes heterotroph. So quite a lot of these dinoflagellates have capability of photosynthesizing, but also have capability of yum-yums, of heterotrophic lifestyle. Sometimes they're even predators. They usually find in coastal waters, warm waters, where there's high amount of nutrients available. They're also very often found as symbionts of benthic and pelagic heterotrophs, so at coral reefs. No, that's where this group is going to be sitting in. The most fame they got as red tide organisms. So when you think about red tide organisms, usually when somebody says red tide, that's what they're talking about. They're talking about these dinoflagellates. And again, this is I'm rolling everything back to perspective, remote sensing and optics. You know, all these things have these important position in ecology of the ocean, important biogeochemical role. So if these are the red tide organisms, you know, you really want to develop a way to study them with optics. So if they're red tide organism, red means that they have some kind of specific pigments that makes them red. So it's something that's rolling it back to optics and allows us to follow them using optics. So this is during my PhD. I did a PhD in Southern California, just in front of LA, and this is Lingolodinium bloom, Lingolodinium polyedra. It's a dinoflagellate. The concentration of chlorophyll is 140 micrograms. So North Atlantic bloom maximum is 5 micrograms. This is so thick, it's like a pea soup. You know, you would, for like the stuff that Colin was talking about, how much would you filter on a filter pad? You would filter like 10 milliliters. It's so thick. This is like red like crazy. So this is a situation where you can see that the pigment is actually, you know, changing the water. So you can use optics to study these kind of things. And these guys are referred to as red tide algae, but they're not toxic. The problem with them is the large amount of biomass, when it dies off, and they all die off because they're going to run out of nutrients, is going to fall on the bottom of the ocean. This in this case really shallow ocean. So what's going to happen then when you have all this biomass falling down? Yeah, then what happens then? Hypoxia. Bacteria comes, yum, 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 yum. Bacteria needs oxygen. No, they don't photosynthesize, so they're going to eat yum, 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 and all the oxygen goes away. And then you have big problems. Then you have fish dies, fish kills, and so on and so on. So things can be harmful not just because they form toxins. And now, the most beautiful things are the diatoms. Now we're like really in a micro world, and there's teeny tinier ones. But these are the big, the beautiful ones. I have the image from the sea lives here showing this bloom in North Atlantic, and there's a really good chance that most of this green here, most of this high chlorophyll here, is associated with the diatoms. There's a gorgeous video that I'm sure Jeremy is going to show. We're going to see the pulsating. It's going to be like earth breathing. That's what Mary Jane once said. And you can see this bloom going up and down, up and down. It's actually following this diatom bloom that happens in the springtime. These are the guys like, you know, like the first flowers of the springtime. They're the most common type of the phytoplankton. They have silica shells. So all these like beautiful things, all these beautiful structures actually built out of silica. They're single cells, but they occasionally build chains. And once again, these chains help them to survive in the environment where they live, either for the nutrient uptake or the fight with the predation or just to stay afloat. Some of them, they have two forms. One of them is pennate, the second one is centric. So a cool thing about diatoms is that if you take a petri dish, I'll explain you know, and everybody knows what a petri dish is. Yeah, more or less. Okay, you're going to see some. They kind of how centric diatoms look like. They look like two petri dishes together, and it kind of helps them later on with the division and reproduction. The pennate one kind of look also like a petri dish, but not really perfect round one, but kind of like more elongated one. And as such, they can form these chains and kind of attach one to another, usually through a process of division. Some of them are toxic, similar to dinoflagellates, and the most famous one is domoic acid. Currently on the west coast we have a gigantic bloom of Pseudonychia. And Pseudonychia is a phytoplankton species, diatom species that forms domoic acid. This domoic acid is it's amnesic, it's a toxin that causes amnesia. So what you have is usually affects mammals and birds. So you have these like crazed birds, and really like in LA and stuff, sometimes like flying in the cars. And the story goes that Hitchcock's movies, Birds, no? So the story goes that he was actually in the seaside in California, and there was like a big, I mean, they can't prove it, but there was a story that there was a big diatom toxic bloom, and there was a fish poisoning, and all these pelicans and birds ate lots of this fish, went berserker, and they were like attacking people and stuff, and that's how Hitchcock got inspiration for his movie Birds. It's in Capitola, California, or something. I think there's actually a paper on that. Anyways, the most important things regarding Hitchcock and their influence on the art and culture of the world is the fact that they're spring bloomers. They're the first thing they're going to bloom. They bloom intensively. They're like yum, 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 most of the nutrients, and then they die. And as they're big, they're going to start clumping up together. They're going to sink very effectively from the surface waters. And therefore, they're going to export all that carbon, most of the carbon with them. And that's kind of like when you talk about climate change and all the potential like ways that we want to remove the CO2 from the atmosphere, that's really important, no? So once again, we really want to be able to study them optically. And in order to do that, we can now use pigments. So everything what I talked about before, it was mostly a morphological structure. Now I'm entering in the field of pigments, and Colin touched a bit upon that, and Mary Jane is going to be talking about more. I'm not sure when this developed, but high-performance liquid chromatography is a method that we use today in order to detect pigments in algae and in other things, but in algae specific. So if you would actually do it yourself, you would take a sample, extract it, blah, 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 run it through the column, and you would get an output similar to this one, where you can see a specific absorption, 450 millimeter, versus this is a retention time in a column. So each of these peaks is a specific pigment. So for example, these are all the pigments that came out from this sample. So here, there's actually 1 and 3 and 4, I think even 2, are different types of chlorophyll C. This is peridinin. This is a diagnostic pigment for dinoflagellates. Here, you can find lutein. Each of these little things, peaks, is actually a pigment. So if you have this, you have your standards, you can say something about the concentration of this HPLC. And as it turns out to be, and this is a very bad image, I apologize, I'll try to find a better one, but it's really, really great because it's showing you pigments versus these groups of algae that I spoke about. And if you look at the size of this dot, tells you something how strongly present the pigment is for each of these groups. So for example, you just have these pigments, and you want to say, okay, is there an easy way to distinguish between red algae and naked flagellates? And you kind of look, it's like, well, yeah, this guy has lutein, this guy doesn't have lutein, this one doesn't have phycocyanin, phycoerythrin, awesome. So I can actually make like a little model in MATLAB and somewhere in Excel and kind of figure out a way how to distinguish these two if I have lots of HPLC samples. And people have done that before. And one of the most famous clustering methods out there is called CAMTAX. It was published in I think it's early 96, and quite a lot of people are using it. So what it's doing is following the assumption that certain species have certain concentrations of chlorophyll, of different pigments. So, stuff that Colin was talking about earlier. Will the chlorophyll concentration change as you move that species up and down in a water column through different environmental conditions? I mean, will the chlorophyll concentration per cell always stay the same? No. And so will not the accessory pigment. So if you do this, be very careful. Very, very careful. You can do it, but you have to kind of fit it for your own area, for the specific area where you work. The same goes for the other clustering method. But it's a great and a powerful tool. And it's very much used in classification in optics because it speaks so nicely to the optical signal. All these pigments have a specific, most of them, expression in optics, and we can use that to collect these two methods. So, you know, taxonomy based on pigments is something that is very, very dear to our heart as optics people. But that's old school. We're now entering like a new era, like last 20 years, where we have DNA. And now when DNA appeared, everything went berserker. So a long time ago, we thought the green algae are like really good friends of red algae, and they have evolved together. And it turns out the green algae, as I said before, are kind of much better friends with oaks than with red algae. So when DNA and molecular biology came to life and started being used as a taxonomical tool, some of the concepts that we had about taxonomy that were based on morphological structures are falling apart. And why? If you think about it, you know, like all these species, kind of like morphology was evolving in a similar way just because of the maybe, you know, the pressures from the environment, the ecosystem niches that they have like kind of, you know, set on. While DNA is showing something else. So with the rise of DNA, you start seeing images like this, and you can like now use DNA to go deeper and kind of look at just eukaryotic cells and how all these different things are distributed. So this is a green algae sitting here. This is where your diatoms are. This is where your coccolithophores are. And this is like next to green algae, this is where your oak is. So there's really more similarity between oak and a green algae than a diatom, which is kind of weird, but that's what it is. And you can use genetics to go even deeper. So all these species that we use to define, so when we say Perchlorococcus, I said once again, we can't use light microscopy to talk to anybody about anything about Perchlorococcus. We can use flow cytometry. But if we start looking at the DNA and actually either expression of a single gene or looking at the whole cell genomics, just single cell genomics, we start seeing all this crazy ass diversity, really great diversity that we couldn't see before. So the question is, I haven't yet to see papers that connect really well molecular biology with optics. There was a great paper from Tara Oceans where both Ali and Emmanuel where there is kind of like they're talking about a little bit about optics, a little bit about DNA, but still those two things have to be combined. And both of them are really powerful tools, and that's where you guys come in. You really have to start thinking a step away from pigments, a step away from classical methodology. Start thinking about molecular tools, what molecular tools can give you, because for example, you can use proteomics here, an expression of RNA, to see what each of these guys is doing, which kind of niche is occupying, and why is it doing that. By combining this data with optics, you're going to have such a great time, such a great data, such a great results. But then we have a newest thing. It's called phytoplankton functional types. And I myself think about it, that's also a taxonomy thing. So finally, just recently, in 2014, we got a definition of phytoplankton functional types. It says this is a group of organisms, irrespective of taxonomic affiliation, so regardless of genetics, regardless of morphological taxonomy, they carry out a particular function, a chemical process such as calcification, silicification, nitrogen fixation, DMS reduction. Functional groups are also sometimes referred to as biogeochemical guilds. This, I'm going to go here, is the sexiest word alive. If you look at the Google Scholar occurrences per year, from 1990 to 2015, look at the growth of that word. It's going up. And it's like, when I was in grad school, harmful algal blooms were the buzzword. And here, how many of you is doing work on PFTs? 1, 2, 3, 4, 5. 5 out of 25, that's a lot, 1/5. So this is the buzzword of the day. And you can see with the increase with development of the finer tools in optics and the increase of the number and complexity of the satellites that we're launching up there, the number of these words are going up. And I know that Jeremy is going to be talking a little bit more about that for sure, but with the preparation for the, I have to change this, with preparation for the new satellites coming up, we're going to be talking about these phytoplankton functional types more and more. So going back, what is that functional type of the phytoplankton? What is that? So going back, it actually combines everything what I talked about before. So, it can be, we're talking about functional type, produces oxygen. We can define functional type based on size or shape. So pico, nano, micro, like round, weird. Diatom weird, not round, not me modeling friendly. It can transport specific nutrients. So I was talking about nitrogen fixers, no? It can precipitate calcium carbon as those coccolithophores. It can play things with silica, so as diatoms or silica flagellates. Has ways to balance to enhance carbon flux, the coccolithophores back again. So there's different things, different roles that each of these phytoplankton groups, phytoplankton functional types can play in the cycling of the elements in the ocean. We can group them up based on their nutritional values to hydrotrophic organisms, based on their being paleo markers, as I was talking about the coccolithophores. We can group them as ability to live in turbulent versus stratified environment. There's so many different ways of grouping them as a functional type. And now all these stories, we're trying to, in order to build optical tools to combine with optics. And then we additionally have this chemical composition. So there's so many different things that we can use to define these functional types. It's a big mishmash out there. And everybody wants to work on phytoplankton functional types. And reason is why? Well, phytoplankton functional types is kind of like a cheaper way to combine morphology, ecology, radiochemistry, and everything. And it's a weird mishmash that we can use like an excuse to say, okay, we're going to do these groups and we're going to try to do them using optics. Why? Because we need more data. Problem is in ecology and everything what we do in biogeochemistry and all this applicative science that we do, where we're measuring biology is always so far behind measurements of physics or chemistry. Like if you think about how fast can we measure physics, what are the scales of measuring physics, it's much, much higher than we can measure biology. So in order to increase the measurements of the biology and knowledge about not just biology, but the chemistry and functionality of that biology, we want to use optics to study phytoplankton functional types. That's why we need more data. And that's where the sexiness of that word is increasing. We can use everything what I said so far to develop these optical tools. We can focus on specific morphological and structure features. So we can use specific pigment structure. As I said, red tide blooms, no dinoflagellates. Then, I don't know, well, there's like phycoerythrin, no? And some of the cyanobacteria and so on and so on. So you can use these specific pigment features. The things that Colin was talking about, the size of the cell will change the way that we see the absorption. So it's again specific pigment structure, or the way the pigments distribute in the cell. Specific size will lead to specific optical signature. And in this case, we talk about particle size class, but as you're going to learn, we can use optics to say something, is it really small, is it medium size, or is it large? So we can go back and divide these particles that we see using optics to pico, nano, micro, and then say something about their functionality. Because once again, in the beginning, I showed you that, for example, most of the DMS producers are sitting in that nano range. So we can then assume something about these particles. We can focus on taxa-specific ecological traits in trophical states. As I told you, the diatoms are the first one who come out, and they're usually the ones who bloom really, really strong, really, really big in the ocean. So when you see in ocean high concentrations of chlorophyll, there's a big chance that's a diatom. So following those assumptions, you can create models that allow you to take certain chlorophyll or IOP measurements, certain chlorophyll concentration, and infer something about the community composition. So these are the two bases of the PFTs. And that's, again, something that Jeremy is going to talk about, and something that you all have to really know a lot about. Why? There's a big chance you're going to be doing that. You're going to encounter that. And the biggest question now, is it feasible to do that? Can we really do that? Now, in this stage of your class, you can't answer that. You're going to learn a lot about how to do proper optical measurements, what these optical measurements mean. So whatever you learn here, take it with you. Take the uncertainties, the pitfalls, the strength, and everything. And when you're going to be making models, when you're going to be making assumptions about phytoplankton functional types, remember, know your friends and know your enemies. Your uncertainties are your friends, but they're also your enemy. Signal-to-noise ratio, things like that. Be realistic in your expectations. There's only so much that you can milk out of 5 wavelengths that come from space. Kurt and Jeremy will talk about atmospheric correction, how much of that is contributing to remote sensor reflectance for space. When you take all that stuff, how much can you actually, with that little bit of reminding signal, can you say about phytoplankton? So be realistic in your expectations and validate. All these morphological things, all these techniques that I talked about and Mary Jane will talk about can be used for validation of your approaches. Remember these 3 things when you do phytoplankton. And kind of done one time, I think I am. And yeah, I am. And so this is the image that Colin mentioned yesterday. So this is the image from a couple weeks ago. This is Russia, Siberia, Alaska. Aleutian Islands are kind of cut off. They're here. Look at this. I mean, this is like Van Gogh went crazy. Like, look at these eddies. I mean, I was staring at this image for like half an hour. There's like, what I mean, like, I'm like, you guys are gonna get to that point of your life, trust me. If you stay on the ocean, or there's an island effect here. Look at this thing. Look at this. There is a mixture of plume, sediment plume. This might be a coccolithophore bloom. We're close to solstice. They usually bloom in. Look at these eddies that they form. Look at these strings here. Look at this is just like amazing. And this is your, this is your biology from space. And this is where you guys are going to be, you will know how to read these things when you're done with this class. So that's it. Do you have any questions? I kind of babbled a lot. I just gave you enough of the concept of phytoplankton. I know that some of you like come from different regions, but it's kind of like a taste of phytoplankton. Okay, well then done. Mary Jane. So Mary Jane's going to kind of take it from here and do it.
5 minutes.
Okay.
And